Okay, welcome everyone to the next iteration of the IIT Theoretical Ecology Seminar. This week we're really happy to have Aaron King from Michigan and Santa Fe. He's going to come and present about exact phylodynamics via structured Markov genealogy processes. Thank you, Aaron. Take it away.
Thanks very much. I really appreciate the invitation to speak. And so I want to talk today about some recent theoretical developments, computational developments that we've been able to make, facilitating improved inference methods for the kinds of models that we care about as theoretical ecologists. So before whoa, what's going on? I've got some kind of a delay here with this. Let me hang on a second here. Okay, some acknowledgments. So a number of people have contributed to this work, including Qianying Lin and Ed Ionides, my collaborators here at Michigan. Qianying is now at Los Alamos. Eric Volz, Simon Frost, and some other folks at various places, as well as some funding that has been instrumental in all of this. So by way of motivation, most of us were aware of the ongoing surveillance for new variants of SARS-CoV-2 during the last several years. And basically that amounts to tracking the number of new molecular variants with sufficient amount of difference through time. And with the availability of cheap genomic sequencing, one can supplement the sort of time series of frequencies of variants with phylogenetic trees that display the patterns of relatedness among the different samples. So I want to talk about that today. Underlying this sort of question of genomic surveillance is sort of how different strains are competing with one another. So in a very cartoonish perspective, we might have in the back of our minds a model like this, where we have some pool of susceptible individuals that are at risk of becoming infected by one of, well, in the simplest case, two different strains. And when they do become infected, they progress through a latent state, an infectious state, and an immune state, perhaps losing their immunity. But the key question would then, of course, focus on the relative transmissibility represented by these two rates lambda, which in turn are dependent upon these two transmission rates, beta 1 and beta 2, as well as potentially other characteristics of the diseases. So we've known for a long time how to use time series data. So every time I touch the mouse, the thing jumps, so you have to be very careful. We've known for a long time how to use time series data of the kind we see here in the upper panel, where we're tracking the number of cases by variant in a particular geographical region through time. We've known how to use data like that to infer the parameters of various kinds of dynamic models for some time. But what I want to talk about today is how we go about using the genealogical or phylogenetic trees that we can extract from data to parameterize models, to perform inference on models. So phylodynamics is the word that's used for this project, and it's a term that is often used in a very broad sense to refer to any kind of inference procedure for any of the determinants of an epidemic spread, of the spread of an epidemic using genomic data. But I want to focus that down during this talk to the specific case where we want to use genomic data to infer the structure and parameters of stochastic dynamic transmission models. So the basic problem, of course, in an inference of inference is to somehow get information to flow from data down to the questions, which in this case, as I say, take the form of a stochastic transmission model. And in this case, one major pathway that one can construct from the data to the model is by way of the tree-like structure, which I'm going to call a genealogy here, that tree-like structure that displays the patterns of relatedness amongst the different genomic samples. Of course, whenever you want to get information to flow in one direction, the proper way to do that is to construct a predictive model or a generative model that predicts what the data will look like on the basis for any given model. And of course, we want that to flow this other way. So we're going to show in this talk how stochastic transmission models give rise to genealogies that can then be related to genomic sequences. So the basic problem mathematically is the following. Suppose we have a set of genome sequences and a genealogical tree that hypothetically relates those sequences. We also have some models. We have typically a rather simple model of sequence evolution along the tree, as well as this model of great interest, the dynamic stochastic transmission model. That's the focus of our efforts. But we may also have some additional time series data, for example, numbers of infections, mortality, vaccine delivery schedules, and so on. And we want to somehow use the data S and Y to infer the properties of E and D, but principally D. So the golden road to doing that, of course, is via the likelihood function. So the likelihood of function of the two components of the model, which is basically the probability density of the data given those models. And one can write that in the following way, where I've factorized it into two components. So if this blue component here, which is the likelihood of the sequence data given the phylogenetic tree and the sequence evolution model, we might call that the phylogenetic likelihood. So Felsenstein showed us one major way to go about computing that. There's been a lot of work done since on how to compute that thing. But what I want to focus on in this talk is this piece in the red, which we might call the phylodynamic likelihood, specifically, which should be phi here, not S. What's the probability of getting a particular genealogical tree and perhaps some other time series data given a dynamic transmission model? Because if we can compute this, and we're willing to follow Felsenstein in terms of his prescriptions for how to compute this, then it's just a simple matter of integrating overall possible trees, he says in a subdued voice, because of course this is a very large space. But nevertheless, while this piece has attracted a lot of attention, an enormous amount of work, this piece has been rather neglected. Okay, so how do we go about doing that now? And how have we done that during the period of the last several years when these phylodynamic methods have been so important in the public health response? Well, there are two basic approaches that have been followed. One is based upon the classical Kingman coalescence. So these models essentially allow one to compute the probability of a given tree by means of a backward in time construction that relies upon the population being very large and the sampling frequency, the probability that an individual gets sampled, is quite small. So these methods, while they're very simple and very appealing, are hard to relate to the kinds of models that we're directly interested in, and especially in the context where we have relatively small populations. For example, when new variants are arising in a population. The second major approach, a little bit more recent, is based upon linear birth-death processes. And because in a linear birth-death process, distinct lineages are statistically independent of one another, one can mathematically write down, this problem becomes tractable, one can mathematically write down expressions for the likelihood. But of course, the models that we're typically interested in are nonlinear, and these linear, so implicitly there's a linearization argument that has to be made in order to be able to apply these methods. So both of these methods imply the need for certain kinds of approximations, and the question I'd like to ask is whether it's possible and what's involved in dispensing with the need for these approximations. So I'm going to do a few things. I'm going to show you first how a given stochastic dynamical population process induces a unique genealogy process, which is a stochastic Markovian process that we can study, how we can project the genealogies that process gives us via a process of pruning and a process of obscuration down onto the space of observable data. And then I'm going to show you how, by putting those two things together, we can derive the exact likelihood of a given genealogy as the solution to a particular nonlinear filtering problem, which can be solved in various ways. So the details can be found in this archive preprint. Okay, so there's a few different pieces. First of all, I need to explain to you what I mean by a population process. And this slide just gives a number of different compartmental model diagrams by way of showing some examples to kind of give you a feeling for what we're talking about. So in particular, we want to have a Markovian process, which is the model, which represents our hypotheses about the generation of the data. And so in the simplest case, that might be something like this SEIR model here, where we have susceptible individuals that have been exposed but haven't yet developed an infection, infectious individuals and immune individuals, or dead individuals potentially. And so these diagrams are relatively standard way of providing shorthand for what these models involve. But they can have lots of different kinds of complexity beyond this, right? For example, I've talked about different strain structure. We might have heterogeneity in the disease progression. For example, this model that has asymptomatic infections as well as symptomatic infections that can lead to hospitalization or death. We might have behavioral complexity. For example, individuals that spread at a low rate and individuals that spread at a high rate. But all other kinds of heterogeneity, including age heterogeneity, spatial structure, physiological structure, and so on, are all possible. The key ingredient that we need here is that the process be Markovian and that the structure be discrete, in the sense that there's a countable number of compartments into which all individuals fall. You'll notice that I've shaded some of these boxes I've shaded some of these, and the boxes that are shaded are a bit special in that individual hosts who find themselves in one of these shaded boxes actually harbor a lineage of the pathogen, right? So these are infected individuals. They may not be infectious, but they're infected. So inside of each individual here is some pathogen lineage, and it's the same obviously here. And that sets these boxes aside from the ones that are unshaded, where these individuals, while they're capable potentially of harboring a pathogen, don't currently do so. So these shaded boxes I'm going to call deems, which is, well, that's what I'm going to call them. And so in this particular model, there's a couple of deems. In this model, there's 4 deems. Here's 3, and so on. This letter D here refers to the set of deems. So we can think about that as just being a countable set of labels. Mathematically, this population process has to be a non-explosive Markov jump process. And I'll use the capital X to refer to that. And such a process, of course, is uniquely specified by 2 things. There's some state distribution at time 0. So what's the probability of finding the system in any particular state? P0 will be the density of that. And then some jump rates. So what's the hazard at time t of a jump from state X to state X prime? These rates should, of course, satisfy some simple, very non-restrictive conditions. And in particular, I note that the theory that I'm going to present doesn't really limit the nature of these jumps. They in particular can be multiple events at any given jump. So I think the most compact way of mathematically summarizing these models is in terms of the Kolmogorov forward equation, sometimes called the master equation in this context. And this is just a statement about conservation of probability. So it says that if we have a quantity W, a density W, that satisfies this equation, and at time 0, is equal to the initial density, then if we integrate that thing at time t over any subset of x, it gives us the probability of finding the system in that state at that time. And as I say, this is just a conservation of probability. This first term here represents all the ways that the system can move from state x prime to state x and integrates all of those up. And this is all of the ways that the system can move from state x to another state, integrates all the probability flux leaving a particular state. And so the Kolmogorov forward equation is a very compact way of saying all the stuff that I've been saying up to this point. The reason I'm presenting this is that we're going to see that the filtering equations we ultimately derive resemble this in certain ways. So to make this a little bit more concrete, here's the population process for this very, very simple SEIR model. Here's the Kolmogorov forward equation. If we look at that a little bit more carefully, well, another way of looking at it is this Markov state transition diagram. So from a given state, we see that there are 5 different ways that the state could change. Either there could be a transmission event, a disease progression event, a recovery, a waning of immunity, or a sample. Each of those corresponds to a particular change of the state. And we can, if we label each of those 5 possible kinds of jumps with a label that we collect in the set U, then the Kolmogorov forward equation can just simply be broken down like this. And if we plug in the expressions that correspond to that particular model, and those integrals all just be reduced to single terms, and we end up with the specific equation, which is the master equation for this SEIR process. So just to connect the abstract terminology, the abstract equations to their specific realization in this context, the point is that for any such model, we can similarly write down such a diagram and write down such a Kolmogorov forward equation, and therefore all such models are being encompassed by the theory that I'm talking about. Okay, so that's what I mean by that's the class of population processes that we're going to consider, these discretely structured Markovian population processes. Now we need to talk a little bit about what a genealogy is. So intuitively, a genealogy is a tree-like structure that relates some perhaps individuals that are extant in the population. So these black dots refer to extant individuals, as well as possibly some samples, genomic samples. And it relates these things to one another in terms of their common ancestry. So 2 different samples here have a common ancestor at this point, according to this diagram, and so on. We can see lots of other structures there. So we're familiar with these things intuitively, but we need to think a little bit more formally about what they are. And so here's some mathematical details that we don't need to get into. The key thing to recognize is that it's possible to encode or formalize the tree structure as a function from a particular function of time. And so the Z here is just a representation of this genealogy structure as a function of time. But there's a little bit more to it, of course. We have, as I say, in all of these models, demes which are distinct from one another, compartments which are distinct from one another, but nevertheless contain pathogen lineages. On the tree, what that means is that at each point in time, every one of these lineages was in one deme or another. And so we can schematize that or visualize that by painting colors on here. So for example, in that SEIR model, if we give the E color, if we give the E deme the color blue and the I deme the color yellow, then we might have something like this, right? Where what this is saying is that at this point in time, this lineage was found in the E deme, and then it progressed into the I deme. There was an infection event here, so here's the I deme, here's the E deme that emerged from that, and so on. So these colored genealogies are also sort of implicit in all of this. And we can, again, more math, but we can represent that coloring as again, a function of time, and therefore, formally, from a formal point of view a genealogy as a triple where it consists of an end time and 2 functions of time that represent the tree structure and the coloring of that structure, respectively. All right, so let's go back to this specific example here with this particular state transition diagram that I showed you before. And what I'm going to show you is that each one of these different kinds of, or really I'm going to assert to you, each one of these different types of events has a particular effect on the genealogy. So let's see what that looks like. So I'm going to break down the total event rate into the event rates associated with each of those different types. and the Kolmogorov-Forward equation just expands this way. And now to see what this looks like, how the genealogy is induced by the population process, let's just focus on a little narrow ribbon of time around some particular event. So it could be any time in here, but it's just a narrow slice of time. And if we look at that, we might see something like this. So each different kind of event in the population process can produce an event, a change in the genealogy. So there are 5 different kinds of events. Here's an event where a transmission has occurred, and a yellow deme individual has given rise to some number, in this case 2 individuals in the blue deme, and that yellow individual has persisted. Here's a death type event where an individual has recovered from infection, in this case, and is no longer present. Here's an event where disease has progressed. So we have a migration type event where an individual lineage moves from the blue deme to the yellow deme. Here's a sample type event where, in this case, 2 samples for some reason were taken simultaneously in 2 different individuals. And we see those samples just here indicated with these blue dots. And then, of course, some events that occur in the population process don't have any change that they make to the genealogy. For example, if an individual host loses its immunity or is a susceptible individual is born into the population, that doesn't affect the genealogy at all. And then, of course, it's possible to have combinations of these kinds of events in various kinds of ways. The point is that once we understand the model and we know what kinds of events we're dealing with, a given population process uniquely induces a genealogy process. So what we end up with is we have the population process X that induces a process G on the space of genealogies, such that the map from X to G is itself random. The key assumption that we're going to make about this process is that lineages within a deme are exchangeable. So while this is not in itself a consequence of the Markovian nature of X, it is very, very much in the spirit of that assumption, in the sense that this assumption is tantamount to saying there's no additional structure in the population beyond that which has been hypothesized in the model. You'll actually hear people say that this is, in fact, a consequence of the Markovian nature of the model, but it's in reality an additional assumption. So let's look at some animations to see what I'm talking about here. I'm going to very carefully not lose my place, which I'm afraid I've already done. What I want to do is pull up some simulations just so we get a sense of what I'm talking about. So here's a simulation of a process corresponding to this SEIR genealogy process. So the black dots represent individuals that are infected at the given time. Let's run that again. And we can see as the infection progresses, their number increases, but they leave behind them a trace of their relatedness. Eventually, this thing dies out, which is why the genealogy eventually disappears. But if we sample that process, so now the blue dots represent samples that are taken, then of course we don't we keep track of the genealogies that relate those samples. So that's what we're looking at here. That you can see what's going on here is as time goes on, new infections occur and give rise to branches in the genealogy. But as individuals recover, those branches are lost, and the genealogy information is lost about things. That information is maintained if we sample. So samples are not forgotten. Here's just another example. This is the classical Moran process that gives rise to the Kingman coalescent. You can see the thing in a little bit, it's quite a bit simpler. and again, what the sample process looks like. So this is the genealogy process that I'm talking about. This is just animations of that, but of course it is mathematically well defined, and it's all explained in the paper how that works. So we've been talking about how what the genealogy process looks like. In some sense, that genealogy contains a whole lot of information, not only all the extant individuals that are in the population in addition to the samples, but also specific information about where in the space of Deems all the lineages were at every point in time. But that's a lot more than we actually can see. In particular, when we actually look at data, we only have data from the samples. We don't know the individuals that have never been sampled. We know nothing about their lineages. And so one can project down from the space of full genealogies onto what I call the prune genealogies by simply throwing away all of the portion of the genealogy that's not ancestral to some sample or other. One can look at the local structure of a prune genealogy in a similar sort of way to what I did before. Here's a simple slice of the genealogy, just like we looked at before. This is the full genealogy here with all of these different lineages. It's in the neighborhood of a point where we have a branching event here, a birth event. Somebody got infected here. When we prune that genealogy, some of these lineages are going to be thrown away because they're not ancestral to any sample. And so just by way of example, perhaps this yellow one was never sampled and these three lineages were never sampled. So we remove them. That's what I'm indicating here with the dashed lines. And we're left with a different sort of genealogy, right? So in this case, yellow moving through, yellow changing into blue, and then here's a blue lineage. In the neighborhood of this point here, which was a migration event, we might prune that away and obtain something, the pruning might result in something like this. And notice that these two things are actually identical, right? They both look exactly the same, even though they correspond to very different full genealogy. So this is a projection, but it's not a one-to-one map. All right, so one can look at certain quantities that are associated with the prune genealogy, namely the number of lineages that emerge from a given event in each Deem, and the total number of lineages that are present in each Deem, and obtain and summarize that information. So as I was saying before, the prune genealogy is specified by two different, is formally identical to having two specific functions in time, Z here representing the tree structure, Y representing the coloring. If we then project, if we throw away the coloring information, we obtain something called the obscure genealogy. So this is meant to be something more along the lines of what we might actually observe. So even though in reality, every lineage at every point in time was present in one Deem or another, when we have no access to that information. So when we look at what we actually have, what we see is just simply the topological or geometrical structure of the tree, not the coloring. And so that corresponds to just these two pieces here. So there's some combinatoric quantities that I'm going to define. I won't spend any time on these details. If you're interested, we could talk more about it. But this little shorthand notation here, this is not a matrix, but really just a product of certain ratios of binomial coefficients. So we'll call it the binomial ratio. And these quantities N, L, R, and S are meant to be quantities that describe the local structure of the population process and the genealogy in the neighborhood. So these are the quantities that we can construct from looking closely at the structure of the tree at any given point. So the first result that we have is sort of one step along the path to the ultimate result we want to have. but it tells us how we can compute the probability distribution of genealogy, of colored genealogies, of prune genealogies. So the question is, what is the likelihood of observing this prune genealogy, assuming we were somehow able to observe it, given the model of the population process? So let's formally state that theorem. So suppose we have a particular prune genealogy Y and Z with some depth T, and I'm gonna write this function phi down here, which is a product of this binomial coefficient and an indicator function, which just is a compatibility indicator. And then the theorem says that if W is such that at time zero, it's equal to the initial density of the population process, and moreover, it satisfies the following equation, then when we sum up over all possible states X at time capital T, we have the likelihood of the full prune genealogy. So let's look at these equations a little bit more. As I said, these are going to resemble the Kolmogorov forward equation, but you notice that the balance has been thrown off. So we have an influx term and an efflux term, but the influx term is modified by this probability phi, which is less than 1. And so this holds at all time points in between events on the genealogy, in between samples, in between branch points, and at events on the genealogy, we have a discrete jump of W. So here, the tilde is referred to the left limit of these functions. So just the value of Y or the value of X just before the current time. All right, so we end up with this filtering equation here, but of course, as I was saying, we never actually see this. We see the topological structure, the branch lengths, the geometrical structure, but we don't see the colors. And so if we're going to actually compute the likelihood of observed data, we need to be able to integrate out across all the possible colors. And so we need a theorem on the likelihood of a given obscured genealogy. And so the solution to this is really going to be a kind of a cheap trick. What we're going to do is we're just going to rely on importance sampling here. We're going to basically propose all possible colorings. And because we know how to compute the likelihood of each of those, we just need to sum that up over all possible colorings. Maybe that sounds reasonable, but as soon as you reflect on how big the space of possible colorings is, it's a non-starter. This is where the representation of the tree as a function of time comes into its own, because turns out we can do this one step at a time across the tree, never having to consider more than the next immediate possible change in the color. So the theorem is a bit tedious to state, but it basically says that if we come up with reasonable, or that is to say, allowable importance sampling kernels, pi and q, and we reformulate the filtering equation instead of moving from alpha and phi to beta and psi here, then we have a filtering equation which formally is the same. It's now got to integrate over possible colorings. This is just colorings at a given point, not colorings over the whole tree. But when we then sum up over all final colorings and all final states, we end up with the likelihood of the given genealogy. So notice that beta times psi here, by definition, because the pi enters here in the numerator and here in the denominator, this is the same equation here. So the only thing, right, this is the same as alpha phi. So the only thing we're doing here is using the importance sampling to integrate over all possible colorings, and doing it in such a way that it can be done sequentially through time instead of all at once. So let's see, what do I do? Yeah, so let's actually look at some examples of this. So we've got some obscure genealogy here. This is what the equations look like. What we have to do is basically, the alpha and the phi are given by the model and the data. So those things are scientifically chosen for scientific reasons. The pi and the q, which relate to the important sampling of colorings, can be chosen, much more arbitrarily. But all they need to do is tell us how we're going to change the coloring at each sequential point. So when we're going to integrate this thing, basically we're going to do it across this tree, moving from left to right. So sequentially in time. This equation turns out to be a strict generalization of both of the existing theories that are currently being used. So in the case of a linear birth-death process, one can compute the likelihood of a given tree and it's in closed form, but one can also integrate it by integrating those equations. And when you do that, you see that you obtain exactly the right result. And the more computational effort you put in, the closer those things become. Here's a slightly more interesting example of a nonlinear process. Here is the filtering equation written down for this SIRS model. So all of those horrible combinatorial things which are written there in the abstract become very simple expressions in the specific case. So this is the adjustment here. In this case, it's relatively simple. There's no important sampling required because there's only one deem, so there's only one color. This makes the thing a bit simpler. So this is the equation that holds in between events at genealogical events, depending on whether they're a branch point, an internal sample, or a terminal sample. We have some adjustment. But here's a hypothetical sampled genealogy, and the likelihood computation here is just a slice across one of the parameters, the parameter that controls the duration of the rate at which immunity is lost. But you can see the curvature, the positive curvature of this tells us that there is information in this tree about this parameter. Finally, let's look at a non-trivial example here where we've got two deems, so still a simple example, but two different deems, fully nonlinear. I won't try to write down the equation here. It gets a little bit long, doesn't fit on the slide so easily. But given a genealogy, this is a relatively small one. Again, we can compute the likelihood using exactly the same Monte Carlo approach here with a very, very naive and simplistic importance sampling scheme. But you can see that, again, there is still positive curvature here indicating that there is, yes, information about this parameter in the data. The increased noise that you see here relative to what we saw before is an indication of is basically Monte Carlo noise due to the fact that we're now integrating over a somewhat larger space than we were in the previous example. All right. So that was a whirlwind tour of the theory. As I say, the full details are here, and we'll have some time to talk about whatever issues you think are interesting in a few moments. But just to kind of summarize, the theory that is contained here and that I've been presenting to you corrects in the sense that it eliminates approximations, and it strictly extends all existing likelihood-based phylodynamic methods that are currently employed. So it is, in some sense, the generalized theory of phylodynamics and the phylodynamic likelihood. It's an exact theory, and so that's interesting. Of course, whether that exactness, what the value of that exactness is in practical applications remains to be seen. Importantly, all the computations that are involved here can be carried out strictly forward in time. And this is really, really important because the kinds of models that we're interested in, nonlinear stochastic models of populations, are not in general cannot be reversed in any kind of meaningful sense, in any kind of they're intractable when reversed in time. And so if one has to consider only models, for example, that have stationary dynamics and reversible processes, that's a very narrow and uninteresting class of models. So this is really allows us to deal with the kinds of models that we find interesting. Unlike the coalescent and linear birth-death processes, the sampling model here is unconstrained. So the process that gives rise to samples can be modeled just like any other aspect of the process, and one doesn't have to make the unsavory choices that those methods require. In addition, because it is a forward in time sequential assimilation of data, existing methods for assimilating time series data that we've known about for a long time dovetail with this perfectly. So there's no difficulty whatsoever in assimilating simultaneously other time series data. Obviously, what we're talking about here is methods for relating trees to stochastic population models. There are many applications of this kind of thing outside of infectious disease epidemiology, and the applications there have yet to be explored. So there are challenges here. I've shown you some simple computations on some simple examples, but there's a great deal more work to be done to really translate these results into scalable algorithms. And so that's a current area that I'm working on, and some other people are working on that with me. But those issues of scalability and computational expense are the key issues, because really what this theory does is translate this difficult problem into essentially a large computation. The key issues, I think, to resolving those issues of scalability have to do with the efficient choice of the important sampling kernel. So doing that in a naive way, like I showed you here, is always possible, but not typically very efficient. And in particular, it's perfectly allowable, according to theory, to borrow information from the future, which should allow one to tailor important sampling kernels quite a bit more efficiently than you can do in a naive way. Then, of course, there are the issues of how one incorporates uncertainty in the tree structure itself, phylogenetic uncertainty, and then lots of fun issues associated with things like reassortment and recombination that we've only begun really to think about. All right, so I think this is kind of interesting from a philosophical point of view a little bit, because we're used to, as population ecologists, as theoretical ecologists, we're used to thinking about populations and how they change, how the structure and size of populations change through time. But organisms are related to one another, and there's always a genealogy that's implicit. And what I'm showing you here is that if we think about how that genealogy itself evolves through time, then we can obtain a deeper perspective on the history of a given process and the nature and magnitude of the processes that shape it. All right, so this is what I've told you about. I don't need to say it again. There are some references here at the back if you'd like to follow them up. But I think we've got some time for questions now.
Great, Aaron. Thanks very much. Yes, if anyone has any questions, the easiest way to do that generally is to raise your hand in the chat bit. It's just click the react button and do it. Are there any burning ones? If not, I've got a couple to take the lead with. Okay, cool. I was sort of saying, I mean, the thing that interests me the most actually came up right at the end of the conclusion when you were saying about links beyond epidemiology, and you sort of teased us a little bit. Like, what kind of scenarios were you imagining it could be applied to?
Well, my mind first went to obviously the kinds of systematics questions to do with the deeper evolutionary history of clades of organisms. I think there's some there. It's it's certainly the case that we have lots and lots of genomic information to ground the trees, and we often have extremely good trees. We can get very good trees. The problem there is, ironically, that the models may not really be all that difficult. There has been in the literature some discussion of diversity dependent speciation and extinction rates, which I've always found kind of mystifying and strange that these kinds of models are considered, because I don't really think of a species as being a real thing. It's not really an actual thing. So there, the models are likely to be simple enough that the interesting models may arguably be sufficiently simple that this fancy technology is not really necessary. But if for some reason one was considering nonlinear models of speciation and extinction, then these methods would be very useful there. More generally, I think, outside of epidemiology represents this kind of interesting place where we have pathogens, some pathogens, not all of them by any means, some pathogens evolve at a rate which is just in the sweet spot so that it's comparable in the timescales are comparable to those of the processes that we're interested in, and not and neither too fast nor too slow. So the information actually accumulates in the genomes about the history of those processes. And what I'm doing here is showing you how we can unpack that information from the genomes. But I think if we were looking at non-pathogenic organisms, that have those same properties, then these methods could certainly apply.
Cool. Thanks. Axel.
So if I have samples only at the tips of the trees, like in the present, and I look into the deep past, is your method any good for this?
Well, there's always the question of whether the data contain information about your question. The method applies, right? The method doesn't make any statement. The theorems don't depend at all upon the nature of the sampling process. In particular, if the sampling happens all at a single moment, this is a special case, right? There will obviously be many questions for which such data will, about which such data will remain silent. They will have no information. Mm-hmm. If there is information relative to a particular question, then I've just shown you how to compute the likelihood, which contains the answer to that, contain everything that the data have to say about that question. So yeah, I'm not making any claims about whether a given model is good for a given data set or anything like that. My observation is just that as scientists, we want to have the freedom to propose models that correspond to our hypotheses, and we would like statisticians to give us the tools that we need to then bring those questions into confrontation with data. So what I've done here is just show you technologically how one can go about doing that.
No, and I like this because it's super fancy and not trivial, but yet kind of there's some interesting math.
Yeah, the math is a little bit interesting. It's true. It's true. It's not trivial. Not entirely trivial. It sort of always looks a bit more trivial in retrospect.
Were there any other questions, anyone about? Because if not, the second thing I was wondering a bit about in terms of the computational challenge with this, do you get a sense of how it scales with the size of the trees that you're dealing with?
Yeah.
So can you legitimately say, like, well, we only need a tree with 1,000 tips or something, and not go for the 100,000s or whatever we've got for COVID?
Yeah, that's a super interesting question. So I don't have. So there's two sort of questions implicit in what you're saying, right? The big question is, right, sort of what's let's answer them in reverse order. So let me share the screen again real quick. So you can see how it scales with the tree size, right? So the way it scales basically is with the breadth of the tree. So if we think about how wide the tree is at any given point in time. So this is a tree which has a very large number of tips, but it's rather ladder-like, right? So that at any given point in time, it's only got a few different branches. So this one might actually be quite a bit easier than a tree that, for example, looks something like this, right? Of course, this is much smaller, but if this had as many tips as this did and that same bushy structure, this would be much more difficult to deal with. So that, the simple question is, how does the computational effort scale with the tree size? But the much more interesting question that you posed was, how much information is needed, right? And clearly, you could easily imagine having very, very large trees, trying to use all the data and bogging down in computational problems in order to get the last 3 decimal points on some parameter estimate, where a much smaller data set would give you enough information. So I don't know how to come up with rules of thumb about that. Obviously, it has to depend, the specific answer has to depend a great deal on the specific system and the specific models that are being used. So it's probably that there's nothing very general that can be said about it. But I think that's a very interesting question.
Cool. Well, thanks a lot. It looks like there's no further questions from unless anyone jump now or forever hold your peace. Okay, I was going to say, yeah, thanks again, Aaron. And for anyone watching or for anyone watching back online, please join us in 2 weeks' time for Jacopo Grilli, who's going to talk about competition, about contingency and functional convergence. Thanks a lot.
Thanks.